For the next part of the digestive system. So here we have a teeth in a longitudinal section to show you the different layers and the different parts of the teeth. So here is a molar. We have this part of the molar that you can see above the gingiva, which is the clinical crown. Below the gingiva, there is this anatomical crown that it is the place where you have enamel in your tooth. So the clinical crown is above the gingiva, while the anatomical crown is everything in your teeth that it is covered by enamel. As we age, this gingiva retracts and you can see more of the crown, so you can see more portions of this visible crown. Now, last time I mentioned to you about the hardness of each of these substances that we have in our body. Enamel is the hardest substance, the most calcified one, the strongest. Second one will be dentin. Dentin has a yellowish color and it's just below the enamel in the crown and below the cementum in the root. And this one is very sensitive to acids, to changes in temperature like cold, hot substances that you have in your oral cavity. And then the third strongest substance that we have in the body will be the bone. So this is the alveolar processes that hold the teeth, and then the teeth are held into the alveolar processes by this periodontal ligament, which is made out of collagen fibers. So sometimes bacteria can come in here in this area, which is the gingival sulcus, and then can penetrate deeper into the root of our teeth, infecting the root, and then destroying this periodontal ligament, and then you end up with losing your teeth. Now, internally to this dentin, we have the pulp, which is an area where you have blood vessels, nerves, and this is the pulp inside the pulp cavity. The nerves and the blood vessels enter through this apical foramen in this root of the teeth. So this is where they perform the root canals, so they remove the pulp when this cavity is infected, and they kill these nerves, and then you have weakening of the dentin and the enamel, so they have to put some reinforcement there with crowns to prevent breakage of this. For caries or dental cavities, so we have normally in our oral cavity, part of the normal microbiota, a lot of bacteria in our oral cavity. But if our diet is rich in processed foods and they contain a lot of sugars in it, bacteria that lives in this oral cavity, they can produce acids as a byproduct of the metabolism of this glucose that they utilize, or these sugars from the foods that we ingest, and that will erode the enamel. So that's not a good idea then to eat a lot of candy, for instance, or processed foods that are rich in sugars. So we have particular bacteria, Streptococcus mutans, that together with other bacteria accelerates the process of producing these cavities. And now the treatment will be removing the damaged area and filling it with either amalgam or ceramic resin. So now prevention will be, of course, to brush your teeth every time that you're supposed to, at least every 12 hours, or 2 times a day for a minimum, and have some coating of your teeth with fluoride. This is an ion that increases the hardness of enamel. And of course, we have we have fluoride in our water to help to prevent cavities. But we have toothpaste that has this fluoride or mouthwash that has fluoride as well. Brushing and flossing is important. What is the jingle that dental hygienists have? Floss the teeth that you want to keep, something like that, it says. So yeah, flossing is very important, not only brushing, but also flossing, and applying these dental sealants that has fluoride to prevent these cavities from happening, and reduce sugar intake. Something that is rich in sugars, or even if it's not rich in sugar, like sugarless sodas. I mean, they are rich in acidity because they have CO2. That also can damage your teeth. Okay, so details about the tongue. So the tongue, it's an skeletal muscle that is covered by stratified squamous epithelium. Intrinsically, you have muscles that helps to move the tongue and give certain shape of the tongue. So the motion of the tongue not only helps to form the bolus, but also helps for articulating words and swallowing the bolus once you form the bolus. Now we have extrinsic muscles that connect the tongue to different bones, like for instance, the one that connects the temporal bone to the tongue is the styloglossus muscle. We have palatoglossus muscle, etc. So these are extrinsic muscles that together with the intrinsic muscles helps to move the tongue, controlling their position. Now, in the surface of the epithelium of our tongue, we have papillae. These papillae contain taste buds. And then not all the papillae has taste buds, but these taste buds help us to detect molecules that give flavor to the things that we ingest. So we have fungiform papillae, which are the most abundant type. They're located scattered throughout the dorsum of our tongue. They look reddish, and they have taste buds. Circumvallate or vallate papillae, they are located between the posterior third of our tongue and the two anterior thirds of our tongue. They are arranged in a V shape. They are the largest papillae, and they also have taste buds. Now, on the sides of the tongue posteriorly, on the edge, we have foliate papillae. These ones are rudimentary, that only when you are a little child, you can have taste buds there that can detect taste, but after childhood, they degenerate and they just become rudimentary without any function. Now, filiform, which are pointed papillae that are also scattered throughout the tongue, and these are the most abundant, they don't have taste buds. However, these are important to detect the texture of food. Texture of food is important. So let's say that someone destroyed these chemicals that give the taste to the food. But with memory, if you already have tasted a particular food, like for instance a piece of apple, if they destroy these molecules that give the taste of the apple, and someone blindfolds you, and then they give you a piece of an apple, and then you will start chewing it and then bring it against your palate, you can tell, yeah, this is an apple, just by texture. So texture is important. Now, in our oral cavity, we have major and minor salivary glands. The major salivary glands are in pairs, and they are large. The largest that are located on the surface of the buccinator muscle, between the skin and the buccinator muscle, will be the parotid glands. Although they are the largest, they are the second most active salivary glands to produce saliva. And we have this duct that drains the saliva from the parotid gland that is called the stensils, or the parotid gland duct, that drains at the second superior molar. This gland can be infected by viruses like mumps virus, and then it causes parotiditis. And the cheeks of the person, they look huge after having parotiditis. Now, something about this mumps virus is that if it infects the parotid glands in a child, this child will have also inflammation of the testicles, and that can lead to damage of the stem cell that produces the sperm cells. So it can lead to infertility when the boy becomes an adult. So then this parotid gland secretes between 25 to 30% of the total saliva. More or less, we produce around 1 liter of saliva per day. Submandibular glands, they are the second largest gland, but it's the gland that it is the most active in producing saliva. It produces between 65 to 70% of the saliva, and it drains on the sides of the lingual frenulum. Not the lingual frenulum, on the sides, yeah, the lingual frenulum. I was thinking in the lip frenulum. So it drains on the sides of the lingual frenulum together with the sublingual glands, and this one, basically together with the sublingual glands, protects the floor of our oral cavity when you produce as much saliva. And this saliva that this gland produces is thick in comparison to the one in the parotid gland. So, by gravity, liquids that we drink goes into the floor of the mouth. So it makes sense that we have more saliva there to protect it against this potential damage to the floor of the mouth when we have drink a liquid that is very hot. And then the sublingual glands, they are inferior to the tongue. They secrete the saliva through several sublingual ducts just under the tongue on the sides of the lingual frenulum. And then makes about 5% of the total saliva. So those were major salivary glands. Now we have minor salivary glands that they don't have a specific name, and then we have thousands of them scattered through our oral cavity. And just as the major salivary glands, they are equally important. If we didn't have these minor salivary glands, our tongue would stick into the palate. We will have very dry mouth, or our lips will stick into the gingiva. I told you in the previous lecture from this chapter that I was going to tell you about the importance of this and how to test it. Tonight when you go home and you will brush your teeth, you're in front of the mirror, do this tiny experiment. Grab a tissue, retract your lower lip, and dry it. And you will see in the mirror. Dry it, and then see how long it takes for the shininess of the internal part of your vestibule to show. If these minor salivary glands, they wouldn't exist in there to basically lubricate your lips against your gingiva. Whenever you speak, you cannot speak because your lips will be all stuck to your gingiva. And the same is for the upper lip. On the inside of our cheeks, we have all these minor salivary glands, and of course we have in the hard palate. So the quality of the saliva varies. So the saliva is produced by the salivary glands with these acinar cells in the inside of these salivary glands, and then these acinar cells can be serous or mucus. The serous secretes a water-based fluid, more liquidy than the mucus The mucus, they have proteins that makes this saliva to be more thicker. So these ones, the mucus cells secrete mucus to continuously moisten this oral mucosa. Actually, most of these minor salivary glands, they have these mucus cells. And then, depending on how many of these cells and which type of these cells the salivary glands have, it is the type of saliva that you will have. Like for instance, the serous cells, which basically secrete this fluid that is rich in enzymes, it is secreted just before or during eat in preparation for digestion of carbohydrates, for instance. So the parotid glands, they have only serous cells. So sometimes when we eat something that is very acidic, like for instance lime juice, I don't know if you have felt it, but your parotid glands contract, feel like a tiny pain in there. They secrete this serous saliva to start neutralizing the pH of this acidity in this lime juice. And then your submandibular glands, they have mostly serous cells and small number of mucus cells. So in this case, the saliva is going to be watery and then more than thick. And then the sublingual glands, they have mostly mucus cells. So the gland that secretes then more thicker saliva is going to be these sublingual glands. So here is the location of these glands. So this is the parotid gland on top of this masseter muscle. And then here's this tensor or parotid duct that empties in this second superior molar. And then here we have the sublingual gland with several ducts found on each side of the sublingual frenulum. And then we have here the submandibular, which is also known sometimes as submaxillary gland, emptying with this duct here together with these sublingual glands. And then if you make a section of one of these glands, like the submandibular gland, you look at it under the microscope, you will be able to see the histology of these these salivary glands. So here is salivary ducts, the largest ones. These are the acinar cells. They are simple cuboidal cells. And these are serous cells versus mucus cells. Mucus cells, you can see them with the mucus inside. Components of your saliva, mostly water. And this water dissolves electrolytes like sodium, chloride, potassium, urea, some waste products can be found in our saliva, and enzymes like catalase, like amylase, salivary amylase. This one breaks down starch so that we can break down these glucose molecules or having digestion of this starch into individual glucose molecules. Lysozyme enzyme, this starts destroying bacteria. Bacteria has a cell wall, so the cell wall is destroyed with this lysozyme. And then secretory IgA, this antibody that protects us against infections. Bicarbonate ions, HCO3-, neutralizes acids from acidic foods. The process of salivation is partly given by the parasympathetic system in an arc. So the stimulus for salivation is someone perhaps describing you food, smelling food, even thinking about food to start the salivation process. If you're hungry, you start thinking, wow, I wanted to go and eat this, or I wish I could have pizza, whatever. You start thinking food, and that's your stimulus. Then that stimulus will be transmitted to the salivatory nucleus in the brainstem by the glossopharyngeal nerve, And then your motor fibers from cranial nerve number 7, the facial nerve, that innervates the submandibular and the sublingual glands, and the glossopharyngeal that innervates the parotid gland, will start the process of releasing acetylcholine into the acinar cells of your salivary glands. So that these ones, the acinar cells, start producing this saliva. Now, we have some drugs like antihistaminic drugs that blocks these acetylcholine receptors in the salivary glands that cause dryness in the mouth. So many of these medications give this warning, well, your mouth is going to be dry. Now, the sympathetic system is similar to the parasympathetic system. However, you will stimulate mostly the secretion of mucous saliva by your sublingual glands. So these other nerves stimulate the submandibular and parotid glands, while the sympathetic system, it is going to stimulate the sublingual glands. Questions about the oral cavity? Now, let's move into the next part of the digestive system, the pharynx. Pharynx, it's a muscular tube that is located posterior to the nasal and the oral cavities. And we have a subdivision, the nasopharynx, that is just posterior to the nose, the nasal cavity. Then we have the oropharynx that is posterior to the oral cavity, and the laryngopharynx that is posterior to the larynx. Both oropharynx and laryngopharynx are lined with stratified squamous epithelium. And in the oropharynx, on the posterior part of the fauces, we have the palatine tonsils, and then at the base of the tongue, we have the lingual tonsils to help to prevent infections coming from the oral cavity. And then the pharynx is a muscular tube with 3 muscles, the constrictors, superior, middle, and inferior constrictor muscles that sequentially contract and relax to funnel the food from the oral cavity into the esophagus. So in order to receive the food, the superior constrictor relaxes, and then once it receives the bolus of food from the oral cavity, contracts, and then the middle pharyngeal constrictor relaxes and then contract, while this one relaxes to receive more food if you have more food in your oral cavity. And then your inferior constrictor relaxes, receives the food from the middle constrictor, and then contracts, and then the food will continue through the esophagus. Actually, the lower pharyngeal constrictor is part of the esophagus too. The esophagus is this tube that is located in the thoracic cavity that transfers the food into the stomach. It measures around 10 inches in length, is posterior to the trachea, and it is lined by the same epithelium as the pharynx, the oral and the laryngopharynx, a stratified squamous epithelium without keratin. And then that's the most internal layer of your esophagus. The next layer is going to be the mucosa and the submucosa, where you have exophageal glands that secretes mucus. Mucus, as any other part of the mucosa, is secreted to the surface of the mucosa to allow the lubrication and the passage, in this case, of food without damaging the most internal layers of the esophagus. The esophagus has 2 layers of muscle, the circular layer and the longitudinal layer, just as the rest of the alimentary canal, with the exception of the stomach. The stomach, how many layers of muscle does it have? I saw this and then this. 3. So it has circular, longitudinal, and oblique. And now, if you subdivide the esophagus into 3/3, the proximal, middle, inferior, the proximal is made out of skeletal muscle, the wall, while the rest is going to be smooth muscle. So up to the superior third of the esophagus, deglutition or swallowing is voluntary. After that, it's going to be involuntary. Once the food goes from the superior third to the middle third, the food automatically will go into our stomach by peristalsis, contraction and relaxation of the smooth muscle cells of these parts. So the upper esophageal sphincter, which is the junction between the pharynx and the esophagus, controls the passage of food between the pharynx and the esophagus. And then at the lower connection of the esophagus between the stomach and the esophagus, you have another sphincter. If you remember, sphincters are circular layers of muscle around an opening that controls the flow of molecules between these sphincters or passage of substances. So at the gastroesophageal junction, we have a sphincter that is called lower esophageal sphincter, also known as cardiac sphincter. This sphincter, if it doesn't close properly all the way, is going to allow the passage of acid from the stomach into the esophagus, into what we call gastroesophageal reflux disease. So that will irritate your esophagus, and if that is not corrected with time, that can lead to esophageal cancer. So during swallowing, both skeletal and smooth muscle cells undergo contraction relaxation, causing peristalsis. So here is the view of the esophagus. So we have a sphincter in here and another sphincter in here. If you magnify the wall of the esophagus, you can see the lumen or the space here, stratified squamous epithelium without keratin, mucosa and submucosa here, and then skeletal muscle cells and smooth muscle cells in the muscularis externa, and then it has an adventitia. Adventitia is the outer layer of these organs of the alimentary canal when they are outside of the peritoneal cavity. Serosa will be inside the intraperitoneal cavity. And then if you magnify a piece of this sphincter in the gastroesophageal junction, you will see the transition between the epithelium. So this is a stratified squamous epithelium, just like this one, which is part of the esophagus, and then you have the simple columnar cells with lobed cells of the stomach. Eventually, if you have GERD, gastroesophageal reflux disease, you can develop something that's called Barrett esophagus. So process of deglutition or swallowing. We have this division of 3 phases: voluntary phase, pharyngeal phase, and then the esophageal phase. So in the voluntary phase, your food goes into the oropharynx by pushing the bolus against the soft palate, and then you will dilate your superior constrictor, your pharynx, and then you will pass the food into the pharynx, initiating the pharyngeal phase. So, in the pharyngeal phase, the passage of the food into the pharynx will lead to the closure of the larynx with the epiglottis. So the epiglottis will fall over the opening of the larynx, preventing the food from going into the larynx. And then this one is controlled by the swallowing reflex that is initiated above the spinal cord in the medulla oblongata when the bolus contacts these sensory receptors that we have in the oropharynx. In the esophageal phase, you start this contraction relaxation of the muscles of the esophagus that leads to the food moving from proximal to distal, reaching this inferior esophageal sphincter, also known as cardiac sphincter. The pressure, it will be sensed of the food by these baroreceptors, helping to relax the sphincter, and then the food goes into the stomach, ending the esophageal phase. Now, we have neurons of the ENS that allows the contraction of the muscles of the esophagus, and then the control of this swallowing is neural. So we have the voluntary phase that is under the control of the cerebral cortex, while the other two phases are regulated in this medulla oblongata by the enteric nervous system. Now, something that happens in peristalsis in general, no matter if it's in the esophagus, jejunum, duodenum, or even the large intestine, is that you have the other two branches of the ANS, which are parasympathetic system and sympathetic system, working for peristalsis. So peristalsis is stimulated by the parasympathetic system, so that increases the contraction of your muscles and relaxation of your muscles, while the sympathetic system inhibits peristalsis. Why we call parasympathetic system rest and digest? Because with these movements, you help to digest the food. And here's the view of the three phases of swallowing. So here we have the voluntary phase, in which you compress the food using your tongue against the hard and the soft palate, reaching the fauces, which is the opening of your oropharynx posteriorly. And then with this compression, you slide the food into the oropharynx. And at the same time, you elevate your soft palate to seal off the nasopharynx. And then that leads to the moving of the food from the oropharynx into the esophagus. And then you start these peristaltic waves to bring the food from proximal parts of the esophagus into distal part. Now, once the food reaches the stomach, you start these propulsion movements of the stomach and mixing waves that help for the bolus of food now be transformed into a smoothie, like a paste that we call the chyme. So the food, once it reaches the stomach, will be mixed with the acids from the stomach and enzymes that we have in the stomach to start digesting proteins and lipids. So this is the gross anatomy of the stomach. The stomach is located in the upper left quadrant of our abdominal cavity, and it's just located below the diaphragm, laterally and anterior to the spleen. And then it has a J shape. So the regions of the stomach will be this dome-shaped region that is called the cardiac region. Then this large portion that is called the body. And then this narrow portion distally that is called the pyloric region. So you have the cardiac region, the body region, and the pyloric region. It has two curvatures. One that is superior, that is called the lesser curvature, and one that is inferior, that is called the greater curvature. Now, the stomach has, in the muscularis, three layers. The inner layer, that is the oblique layer, the middle layer, which is the circular, and the outer layer, which is the longitudinal, and the epithelium, which is simple columnar epithelium with lobed cells. It is an epithelium that has specific cells, like the parietal cells that produces hydrochloric acid. And then we have mucus cells that produces mucus. And then we have chief cells that produces other molecules. So we will see each of those. Now, the folding of the epithelium inside the stomach, it is great when your stomach is empty, and these folding are known as rugae. Wrinkles in Spanish are called arrugas. So inside our stomach, when the stomach is empty, you have rugae. And then when the stomach is full, expands and temporarily stores the food, so that rugae disappears. And now the stomach connects to the liver in the lesser curvature by the lesser omentum, and then it has in its greater curvature this apron-like tissue that hangs from there, that it holds lipids, so this will be the greater omentum. And now, in this region, which is the pylorus, you have a canal that is the pyloric canal that leads into this second sphincter from the stomach, which is the pyloric sphincter. That controls the passage of the chyme into the first part of the small intestines, which is the duodenum. So if you magnify the wall of the stomach, you will be able to see the epithelium, which it is here in the mucosa, in this layer. And then within this epithelium, we have a gastric pit that leads to this gastric gland. And then underneath this epithelium, we have the submucosa with blood vessels that provides blood supply to this epithelium indirectly, oxygen and nutrients. And then underneath, we have, from this submucosa, we have this muscularis externa with the 3 layers, and then we have the serosa. And somewhere around this area, it's not shown here, we have MALT, mucosa-associated lymphoid tissue. In the pyloric region, a bacteria that can resist acid can live. This is Helicobacter pylori. If someone has chronic infection with this bacteria causes first gastritis, which is inflammation of this mucosa, then can cause an ulcer, and that ulcer can deepen and then can rupture. But as well, these bacteria can inactivate tumor suppressor genes, and then that can lead to cancer of the stomach. Now, we're going to magnify these gastric glands and then this area, so you can see the components of the cells of the epithelium. So if you magnify that region, then you end up with these cells of the gastric pit, and in the neck of this gastric pit, or this area, that's where you have mucus cells that secretes the mucus together with these superficial mucus cells in this superficial area. Now, here in this gastric gland, we have these parietal cells, these chief cells, and we have this bottom part where you have these other cells. And then for the parietal cells, these cells in here, they secrete hydrochloric acid. And then we have within this region a potassium hydrogen pump that uses ATP that helps to bring hydrogen ions into the lumen of this gland so that you can combine these hydrogen ions with chloride that diffuses across these cells to form HCl or hydrochloric acid. Why we don't make hydrochloric acid within these cells? Well, because this hydrochloric acid can destroy these parietal cells if we don't do it like that. Now, the chief cells secretes a precursor of an enzyme that is called pepsin. That precursor is known as pepsinogen. So pepsinogen can be converted into pepsin when you have hydrochloric acid. So this is a proenzyme that needs hydrochloric acid to become pepsin. And pepsin helps to digest proteins. So the proteins can become polypeptides with this pepsin. Now, in the bottom of this area, we have DNS cells that are also known as G cells. These G cells secrete a hormone that is called gastrin. The gastrin hormone stimulates the secretion of these parietal cells and also stimulates the movement of the stomach so that the stomach can mix the food. So gastrin goes into the bloodstream together with other hormones, and then you can stimulate the movement of the stomach. Now, the parietal cells also secretes a factor that is called intrinsic factor. The intrinsic factor codes vitamin B12, so the vitamin B12 can be absorbed in the ileum, the last part of the small intestine. If someone has gastritis, if someone is an alcoholic, these cells will be damaged. You cannot protect your vitamin B12, and then you end up with deficiency of vitamin B12, which is called pernicious anemia. If someone has a tumor in the ileum and they remove the ileum, the person won't be able to absorb vitamin B12, and they also will have pernicious anemia. There is some antibodies that can destroy these or attack these cells, and you don't produce enough intrinsic factor, so you end up with pernicious anemia. What is the treatment for this anemia? Giving shots, in this case, of vitamin B12. So now our hydrochloric acid, it is secreted during the 3 phases. So we have of digestion. We have the cephalic phase, which is basically when you smell, when you see, or when you have stimulating thoughts by thinking about food. That will stimulate your vagus nerve. Your vagus nerve is going to stimulate your DNS cells or G cells, so you secrete gastrin, and that gastrin stimulates your parietal cells to secrete hydrochloric acid. And then also your G cells will secrete histamine that stimulates the hydrochloric acid release or hydrogen release, so you can combine it with chloride. And then you have a negative effect by these G cells into the secretion of a hormone that is called somatostatin, so that you can keep these cells releasing hydrochloric acid. And then your vagus nerve can stimulate directly these cells too. Now, during the next phase of digestion, which is the gastric phase, when your food, if you were lucky, your food goes into your stomach, your vagus nerve and your ENS stimulates more secretion of hydrochloric acid, because that's when you receive food. And now certain foods stimulate more secretion of hydrochloric acid, like proteins, for instance, or the presence of amino acids. And then you will have a positive feedback. Your gastrin will stimulate more your G cells, and then you will produce more hydrochloric acid. And similarly, your G cells will prevent the secretion of somatostatin. So you can continue the cycle. Now, once you finish your gastric phase, the food will be released little by little by opening this pyloric sphincter. So you will release like around 150 milliliters of the stomach contents into your duodenum, and that is going to contain partially digested proteins. So you have polypeptides going into your duodenum. That will stimulate your vagus nerve briefly, and then that is going to stimulate also some G cells that we have in the intestinal epithelium. That is going to lead to the release of gastrin, intestinal gastrin. That is going to release more hydrochloric acid. Now, if you have declining pH and the presence of lipids in your duodenum, as part of the chyme, the content that goes into your duodenum, you will release two hormones, secretin and gastric inhibitory peptide. This will prevent the release of more hydrochloric acid by your cells. And then the presence of lipids, again, and declining pH, like if you have acidity in your duodenum, is going to also lead to the basically the negative feedback on your enterogastric reflex. So that is going to prevent your activity of your vagus nerve and hydrochloric acid secretion, because you don't need to secrete more acid when you have your stomach empty. Now, your stomach, with the presence of gastrin, is going to have different types of movement. So your stomach is going to have contraction of these smooth muscle cells that is going to start the propelling of small amounts of chyme into your pyloric sphincter, helping to open this pyloric sphincter. Then the rest will be propelled back. So these are propulsory movements, and then you will have contraction of your muscle cells in your stomach, the circular layer and the longitudinal layer, plus the oblique layer, making churning movements. So these are peristaltic waves that are going to help to produce this chyme. And then the remaining of the chyme is again propelled against the sphincter, opening the sphincter, leading to more release of the chyme so that you can empty the stomach. The emptying of the stomach varies depending on the type of food that you ingest. Solid foods take longer to be processed and then emptied than liquid foods, of course, and then proteins take longer to digest than carbohydrates and lipids. Now, for the gross anatomy of the small intestine is subdivided into the first part, which is the duodenum, which is small in comparison, of course, to the jejunum, which is the second largest or longest, and the ileum, which is the largest part of the small intestine. Now, these intestine loops are held into the posterior wall of the abdominal cavity by the mesentery. And then the ileum ends in the ileocecal valve, which is an sphincter that prevents feces from the cecum of the large intestine to go and back up into the ileum. For the wall of the small intestines, so we have an epithelium, which is simple columnar cells with microvilli. This microvilli has enzymes that are called the brush border enzymes that helps to break down lipids, carbohydrates, and proteins. And within these cells internally, we have the capillaries, the lacteals that absorb the nutrients and the water that are in the lumen of these intestines. Now, there are foldings in the intestines that increases the surface area so that the intestines absorb more together with this microvilli because that increases the surface area for absorption. But instead of calling these rugae, these foldings, you call them plica circularis, or circular folds. And then underneath this epithelium, we have some mucosa, and then we have as well the longitudinal layer of muscle cells and the circular layer that lead to peristalsis. And then we have the serosa, which is the last area. Now, this is the view in the microscope of these intestinal cells. Simple columnar epithelium again with lobed cells. This is the peristalsis and segmentation movements that happens in the intestines. So when you contract the circular muscle, you transfer the food from proximal areas to distal areas. And then when you contract the longitudinal muscles, you have greater movement of these intestinal contents to a longer area. When you're brushing your teeth, if you're running out of toothpaste, something that you do is you squeeze it, the part where you see that you have toothpaste, and then you slide it, right, to move this toothpaste toward the opening of the container. So that's basically the contraction of the circular muscles when you squeeze it like this, and then when you slide it, will be like if you were contracting the longitudinal muscles so that you can propel your toothpaste. And then, so this is peristaltic movements, and then you have some relaxation within these longitudinal muscles, and then contraction of your circular muscles. That leads to have something that is called segmentation. So you have areas where your food will keep mixing, but in this case, it's not to make a paste. It's actually to mix it with the enzymes that you have in your brush border so that you can digest better the food. So here is a table showing you the action of these different enzymes and molecules. Not all are enzymes. So gastrin, this one partially digests proteins by, and it's released, when you stimulate your vagus nerve, increasing the acid production by your parietal cells. So it's an indirect effect. This is a hormone, it's not an enzyme. So gastrin stimulates the secretion of acid so you can digest your proteins. Histamine is released when you stimulate your vagus nerve, increases acid secretion by your parietal cells. Serotonin, this one is secreted when you distend your stomach. It stimulates the motility of your stomach. Somatostatin has the opposite effect, decreases the stomach pH, decreasing acid secretion by your parietal cells. Hormones that have effects on different parts of the digestive system. For instance, we have cholecystokinin or CCK, that it is stimulated when you have partially digested proteins in your stomach, or you have lipids coming into the duodenum. So this CCK is going to make your gallbladder to release bile so that you can emulsify these lipids so that your big droplets of lipids can become smaller. and then with the bile, and then your pancreatic enzymes can digest these lipids. Now, this one stimulates as well the secretion of pancreatic enzymes and relaxes an sphincter that is called the hepaticopancreatic sphincter. GIP is stimulated the secretion, gastric inhibitory peptide by the entering of chyme into your intestines. This one inhibits the secretion of acids from the parietal cells. Intestinal gastrin, when your chyme enters into your small intestines, stimulate the secretion of acid by your parietal cells. Motilin is released during regular fasting, stimulates the migrating motor complex of the small intestines. So you have a lot of peristaltic movements, and then sometimes you can hear it. This is called borborygmus. Secretin, it is released by the presence of digestive proteins in your duodenum, inhibits the motility of the movement of your stomach. So it is opposite to gastrin. And also inhibits the secretion of acid, stimulates the release of bicarbonate, because you need to protect your duodenal cells from the acidity coming from the chyme. So this is secreted by duodenal cells and increases the production of bile from the liver. Now, BIP, or vasoactive intestinal peptide, is released by the cells of the intestines when you have partially digested proteins coming into your duodenum, inhibits the secretion of acid by your parietal cells, stimulates the pancreatic secretion of proteases to digest these proteins, and increases intestinal blood flow. That's why it's called vasoactive, because your blood flow increases. Any questions so far? Now, for the large intestine, it has this region that is called the cecum. This is the first portion. Next to the cecum, you have this extension that is called vermiform appendix. And then we have here the ileocecal valve. And this part that goes up is on the right side of our body. This is called the ascending colon. Then the ascending colon gives a turn, which is called hepatic flexure, and then runs from right to left, forming the transverse colon. And then the transverse colon gives a turn down into what we call the splenic flexure, and then this part is going to be descending colon. It's on the left side of our body. Then you give an S-shaped turn. This is going to be called the sigmoid colon, and then you will have a straight portion, small portion, that is the rectum, where you store temporarily the feces, and then you have the anus, and you have an internal anal sphincter, which is made by smooth muscle cells, and external anal sphincter, which is made by skeletal muscle cells. External anal sphincter is voluntary, internal anal sphincter is involuntary. Now, something that you can see in the surface of these loops of large intestine is the presence of outpocketings. These are called astras. And then you have a smooth muscle longitudinal layer that is very thin, that forms like a band, that is called the tenia coli. And then you have hanging tissue of lipids, fat tissue, that is called the epiploic appendages. So, in the anal canal, we have grooves that are called anal columns. And then in there, we have anal sinuses that secrete mucus. And you have a transition of epithelia between this area at one line that is called the pectinate line, which is this. So over here, the epithelium is going to be simple columnar with globed cells, you have abundant, in the submucosa, abundant glands that secrete mucus to lubricate this area, because this food that is temporarily stored in the rectum is very hard. So you want to have smooth passage of the food outside of the body passing into this epithelium, and then you have the epithelium of the skin, which is a stratified squamous epithelium with keratin. So these are the anal columns, and then in here we have the internal anal sphincter and the external anal sphincter. And then for the defecation reflex, so as you accumulate undigested food, feces here in the rectum expands. That leads to the release of action potentials by these sacral nerves that initially have a short reflex. So these are parasympathetic nerves. And then this short reflex is going to lead to the efferent signals from these nerves, part of the parasympathetic system, to contract the wall of the anus and the rectum. Now, eventually, when this pressure increases because you accumulate more feces in this area, that leads to a long reflex that is going to travel into upper areas of your spinal cord and then to your cerebral cortex. And then you will have an effector or an efferent signal that is going to lead to relaxation of the internal and external anal sphincter, and then you have defecation. Accessory glands, the pancreas. The pancreas, it has like a hammer shape. This is the head, this is the body, which is the largest part, and it has a tail. This tail of the pancreas is intraperitoneal, while the rest is extraperitoneal, retroperitoneal, I should say. The pancreas is a mixed gland, so it's part endocrine, part exocrine. The exocrine portion, which is 99% of the tissue, releases enzymes that digest lipids, proteins, and carbohydrates. And these enzymes are made in this acini. So these are the acini from the pancreas that connects into ductules that forms this pancreatic duct, the main pancreatic duct that empties into this second portion of the duodenum at the hepatico-pancreatic papillae or major duodenal papillae. So these enzymes, together with bicarbonate, will be released by the pancreas. And then we have the common bile duct, which is part of the duct that drains the bile from the liver into this same portion. And then we have an accessory pancreatic duct that releases also this secretion for the pancreas, just in case that this one is obstructed, you have this accessory pancreatic duct. And then the rest of the pancreas tissue, 1% is made by the islet of the pancreas, or islet of Langerhans, that releases glucagon, somatostatin, and insulin to control the levels of glucose. So we will stop here